Chapter 10 Cell Cycle and Cell Division. During the division of a cell, DNA replication and cell growth also takes place. True. 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 Cell growth in terms of cytoplasmic increase is a discontinuous process. False. 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 DNA synthesis occurs only during one specific stage in the cell cycle. True. 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 The human cell divides once in approximately dash hours. 24 hours. 24 hours. 24 hours. Each can progress through the cell cycle in only dash minutes. 80 minutes. 90 minutes. 90 minutes. Identify the phase of the cell cycle shown in diagram 1. Diagram 1 X interphase, Y M phase, P G0 phase, Q G1 or S phase, T G2, A prophase, B metaphase, C anaphase, D telophase, E cytokinesis. Diagram 1 X interphase, Y M phase, P G0 phase, Q G1 phase or S phase, T G2 phase, A prophase, B metaphase, C anaphase, D telophase, E cytokinesis. Diagram 1 X interphase, Y M phase, P G0 phase, Q G1 or S, T G2, A prophase, B metaphase, C anaphase, D telophase, E cytokinesis. Cell cycle is divided into two phases. Name them: interphase and M phase. Interphase and M phase. Interphase and M phase. The interphase lasts more than %% of the duration of cell cycle. 95%. 95%. 95%. M phase is divided into karyokinesis and cytokinesis. Karyokinesis and cytokinesis. Karyokinesis and cytokinesis. Interphase is also called dash phase. Resting. 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 In interphase, cell is preparing for division by undergoing both cell growth and DNA replication in a disorderly manner. False. In an orderly manner. False. In an orderly manner. False. In an orderly manner. Interphase is divided into three phases. Name them: G1 phase, S phase, G2 phase. G1 phase, S phase, G2 phase. G1 phase, S phase, G2 phase. Dash phase corresponds to the interval between mitosis and initiation of DNA replication. G1. 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 Slight DNA replication happen in G1 phase. False. 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 DNA synthesis takes place in dash phase. Yes. 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 There is an increase in the number of chromosomes in S phase. False. 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 In S phase, central duplication begins in nucleus. False. 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 If DNA is 10 pg in a cell, after S phase it will become 20 pg. 20 pg. 20 pg. What happens in G2 phase? Proteins are synthesized in preparation for mitosis. Cell growth continues. Proteins are synthesized in preparation for mitosis. Cell growth continues. Proteins are synthesized in preparation for mitosis. Cell growth continues. Onion have dash number of chromosomes. 16. 16. 16. Heart cell do not divide. True. 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 Cells after completing M phase enter first into G0 phase. False. 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 G0 phase is also called dash. Quiescent phase. Quiescent phase. Quiescent phase. Cell enters G0 phase by exiting dash phase. G1. Exiting dash phase. G1. 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 Cell entering the G0 phase exit the cell cycle. False. 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 Haploid cells never divide through mitosis. False. 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 Dash is the most dramatic period of the cell cycle. M phase. M phase. M phase. It is also called dash. Equational division. Equational division. Equational division. Cytokinesis is a part of M phase. True. 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 Cell division is a progressive process and very clear-cut lines can be drawn between various stages. False. 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 Name the 5 phases. Prophase, metaphase, anaphase, telophase, cytokinesis. 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 Prophase, metaphase, anaphase, and telophase. Identify the stage of mitosis shown in diagram 2. Diagram 2, A. Anaphase, A. Anaphase, B. Late prophase, B is late prophase, A. Anaphase, B. Late prophase, C. Transition to metaphase, D. Telophase, E. Early prophase, F. Interphase, G. Metaphase. A. Anaphase, B. Late prophase, C. Transition to metaphase, D. Telophase, E. Early prophase, and F. Interphase, and G. Metaphase. A. Anaphase, B. Late prophase, C. Transition to metaphase, D. Telophase, E. Early prophase, F. Interphase, G. Metaphase. A. Anaphase, B. Late prophase, C. Transition to metaphase, D. Telophase, E. Early prophase, F. Interphase, G. Metaphase. The two asters together with spindle fibers form mitotic apparatus. Mitotic apparatus. Mitotic apparatus. Key features of metaphase is spindle fibers attached to kinetochore, chromosome align along metaphase split. Spindle fibers attached to kinetochore, chromosome align along metaphase split. Spindle fibers attached to kinetochore, chromosomes align along metaphase split. Key events of anaphase are centromere split and chromatic separate. Chromatids move to opposite poles. Centromere split and chromatid separate. Chromatids move to opposite poles. Centromere split and chromatid separate. Chromatids move to opposite poles. Nucleolus, Golgi complex and the endoplasmic reticulum reforming telophase. Dash serves a site of attachment of spindle fibers. Kinetochore. In S and G2 phases, the new DNA molecules formed are not distinct but intertwined. True. Condensation of chromosome is completed in prophase. False in metaphase. Dash begins to move towards opposite poles of the cell in prophase. Centrosome. What marks the start of the second phase of mitosis? Complete disintegration of the nuclear envelope. Chromosomes are clearly observed in which stage, that is, morphology is studied in metaphase. They are thickest and shortest here. Two daughter nuclei are formed in dash phase. Telophase. Each centrosome radiates out microtubules called asters. Disc-shaped structure at the surface of the centromere are called kinetochore. The plane of alignment of the chromosomes at metaphase is called metaphase split. Dash is marked by the initiation of condensation of chromosomal material. Prophase. Migration to the opposite pole starts in dash. Anaphase. Cell at the end of prophase don't show Golgi complex, endoplasmic reticulum, nucleolus and nuclear envelope. Golgi complex, endoplasmic reticulum, nucleolus and nuclear envelope. Golgi complex, endoplasmic reticulum, nucleolus and nuclear envelope. Chromosomes decondense and lose their individuality in which phase? Telophase. The end of prophase is marked by which characteristic events? Chromosomal material condenses to form compact mitotic chromosome. N. Formation of mitotic spindles. Chromosomal material condenses to form compact chromosomes. Second one is formation of mitotic spindles. Chromosomal material condenses to form compact mitotic chromosomes. Second one, formation of mitotic spindles. It is most easily studied in metaphase. Morphology of chromosomes. Cytokinesis. In an animal cell, this is achieved by the appearance of a dash furrow. In animal cells, cytokinesis occurs centripetally or centrifugally. Centrifugal cytokinesis occur in plants. Plant cell cannot undergo cytokinesis like animal cells because presence of relatively inextensible cell wall. Formation of the new cell wall begins with the formation of a simple precursor called dash cell plate or phragmoplast. Cell plate represents the dash between the walls of two adjacent cells. Middle lamella. In some organisms, cytokinesis do not occur, forming dash example dash. Syncytium example liquid endosperm of coconut. A very significant contribution of mitosis is called cell repair. True. Meiosis ensures the production of diploid phase in the life cycle of sexually reproducing organisms. False. Haploid phase. Only a single cycle of DNA replication happens in meiosis. True. Recombination between sister and non-sister chromatids of homologous chromosomes happens in meiosis. Non-sister. Identify the stages in meiosis 1 shown in diagram 3. A. Anaphase 1. B. Metaphase 1. C. Prophase 1. D. Telophase 1. A. Metaphase 1. B. A. Anaphase 1. B. Metaphase 1. C. Prophase 1. D. Telophase 1. A. Anaphase 1. B. Metaphase 1. C. Prophase 1. D. Telophase 1. Prophase 1. 65. Prophase of the first meiotic division is typically shorter and less complex when compared to prophase of mitosis. False. Longer and more complex. The 5 phases of prophase of meiosis 1 are leptotin, zygotin, pakitin, diplotin, diakinesis. Paired chromosomes are called dash chromosomes. Homologs. The first 2 stages of prophase 1 are relatively long-lived compared to pakitin. False. Short-lived. By the end of dash, nucleolus disappears and the nuclear envelope dash also breaks down. Diakinesis. Chromosomes becomes gradually visible in leptotin. Chromosome synapsis is accompanied by the formation of complex structure called dash synaptonemal complex. The complex formed by a pair of synapsed homologous chromosomes is called a dash or dash, bivalent or tetra. bivalent or tetrad. The compaction of chromosomes continues throughout dash stage. Reptotin. Chromosomes are fully condensed in which phase? Diakinesis. Packeting is characterized by appearance of recombinant nodule. What is a recombinant nodule? The site at which crossing over occurs between non-sister chromatids of the homologous chromosome. The site at which crossing over occurs between non-sister chromatids of the homologous chromosome. The site at which crossing over occurs between non-sister chromatids of the homologous chromosome. Enzyme involved in crossing over is recombinase. Recombination between homologous chromosomes is completed by the end of dash. Packeting. Beginning of diplotin is recognized by dissolution of the synaptonymal complex, tendency of homologous chromosomes of the bivalents to separate from each other except at the sites of crossovers. Dissolution of the synaptonymal complex, tendency of homologous chromosomes of the bivalents to separate from each other except at the site of crossovers. Dissolution of the synaptonymal complex, tendency of homologous chromosomes of the bivalents to separate from each other except at the site of crossovers. Dash represents transition to metaphase. Diakinesis. In dash, diplotin can last for months and years. Usage of some vertebrates. Dash is marked by terminalization of chiasmata. Diakinesis. 4 chromatids of each bivalent clearly appear as tetrad in packeting. Chromosomes start pairing together in dash stage, and this process is called dash. Zygote synapses. X-shaped structures called are formed in chiasmata diplotin. Metaphase 1, anaphase 1, telophase 1 and meiosis 2. Metaphase 2, anaphase 2, telophase 2 and meiosis 2. Identify the stages in meiosis 2 shown in diagram 4. Diagram A, metaphase 2, B, telophase 2, C, prophase 2, D, anaphase 2. A, metaphase 2, B, telophase 2, C, prophase 2, D, anaphase 2. A, metaphase 2, B, telophase 2, C, prophase 2, D, anaphase 2. Bivalent chromosomes align on the equatorial plate in dash. Metaphase 1. In anaphase 1, sister chromatids separate. False, homologous chromosomes separate. Chromosomes move towards opposite poles by shortening of microtubules attached to kinetochores. No replication of DNA occurs during interkinesis. True. Diad of cell is formed in telophase 1. In many cases, the chromosomes do undergo some dispersion in dash phase. Telophase 1. In telophase 1, they reach the extremely extended state of the interphase nucleus. False. Dash are very important for the process of evolution. Variation. Diad of cells are formed at the end of telophase 2 as it resembles telophase of mitosis. False. Tetrad of cell. Meiosis is the mechanism by which conservation of specific chromosome number of each species is achieved. True. Karyokinesis is divided into prophase, metaphase, anaphase and telophase. Which stage involves major reorganization? M phase. M phase. M phase. Each centromere radiates out microtubules called asters. 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 Shape of chromosomes is studied in dash stage. Anaphase. 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 S phase marks the phase of DNA replication and chromosome duplication. False. Only DNA replication. False. Only DNA replication. False. Only DNA replication. Period of cytoplasmic growth is G2 phase. G2 phase. G2 phase. In early prophase, chromosomal ends are fused with nuclear membrane. True. 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 Interkinesis is short or long-lived? Short-lived. 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 4 haploid cells are formed at the end of meiosis II. True. 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 In telophase 1, nuclear membrane and nucleolus reappear. True. 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 Which stage of meiosis involves Which stage of meiosis involves division of centromere? Anaphase 2. Anaphase 2. Anaphase 2.